This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 52, recorded on March 7th, 2013. Hi, everyone. I'm Vincent Racaniello, and you're listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from the Medical University of South Carolina, Michael Schmidt.
Hello, Vincent.
How are you?
I'm doing really well. It's a little bit nippy here for South Carolina, but it's far better than with the weather I understand you're getting up on the East Coast. It sounds like Hurricane Sandy Part 2.
No, we haven't had anything here. It's a bit windy, but no snow. It was supposed to snow, but we have nothing.
Oh, well, that's good, I suppose.
How was your caving adventure? Did you go caving after our last episode?
I wanted to, but I didn't.
Okay. We have a special guest today. She's joining us from California, from Stanford University Medical Center. Ellen Jo Barron, welcome to the show.
Thank you, Vincent.
You are our first clinical microbiologist.
Well, that's sad. There are so many of us, and infectious disease is so important in the world.
I know.
But I'm glad to be here.
Well, maybe you'll start a trend, and we'll have more on once. You have to be a pioneer, I guess, and that's what we wanted to do. We wanted to get you on today because you've been in the field for many years, and we hope you can tell us about what it is, what you do, how you get there, and all those sorts of interesting things.
Well, I'd be delighted. And as a point that you made about the many years, since I have been a clinical microbiologist, we have recognized new etiologic agents of infections such as Campylobacter species and Legionnaire's disease and SARS and HIV and, my goodness, so many new things that it's really an exciting career. And of course, there's a lot of career stability and longevity because there's always some new infectious agent that we have to figure out how to detect and treat.
Well, to go into the current events that are spreading like wildfire across the United States, I'd like to gauge your thoughts on CRE, the carbapenemase-resistant Enterobacteriaceae. I guess my principal question is, why now?
I think those organisms have originated in Southeast Asia, and it's sort of interesting because we've seen them in the United States here and there. There were some episodes on the East Coast a couple of years ago. But I think, for one thing, laboratorians were not necessarily doing all the tests necessary to recognize them. But secondly, I think there seems to be more of them coming from Southeast Asia and India, and we have medical tourism now and people going into hospitals over there and then coming back here. And those are some of the cases that we're seeing, is that even a patient who's a child who stayed one day in a hospital in India came back having become colonized with New Delhi metallo-beta-lactamase producing Klebsiella, which is one of the most common carbapenemase resistant organisms. So I think it's just a conglomeration of effects that we're beginning to see.
I wonder if we could step back a bit and first, Ellen Jo, understand how you came to be in this field. I know you have a PhD, is that correct?
Yes, that's correct.
And where did you get that, and in what field was that?
I received my PhD in medical microbiology and public health at the University of Wisconsin-Madison.
Wow, the hotbed of bacteriology.
Yes, and Howard Timmon, a Nobel Prize winner, was one of my early professors.
Wow.
As a matter of fact.
Did you do your research in someone's laboratory there?
Yes, I was the very first student of Dr. Richard Proctor, and he was one of the few MDs, actually maybe the first MD in the Department of Medical Microbiology at Wisconsin, and he was interested in Staphylococcus, but I did my PhD in Salmonella typhimurium, and it was very interesting and groundbreaking research, which had difficulty being published because it went against the current thinking. And what we did was compared in vitro neutrophil activity of mouse neutrophils against the mouse typhoid organism Salmonella typhimurium, and then human neutrophil activity against the human typhoid agent Salmonella typhi. And my work showed that they were radically different early responses. And nobody wanted to buy that because many of the premier researchers in the field had been using the mouse model of typhoid, looking at vaccines, et cetera. And I was showing them that it was an inappropriate model. And I think nowadays everybody agrees that there are all kinds of problems with certain animal models with regard to the innate immune system. But when I found it back in the 70s, it was a hard sell.
So it sounds like you were doing basic microbiology research. Did you, at that time, did you envision a career in clinical microbiology?
No, but it's an interesting topic because I had been a clinical microbiologist, a bench tech microbiologist with my bachelor's degree for 12 years before I went back to graduate school.
Oh, I see.
And I went back to graduate school specifically because I felt that I had something important to say to physicians. And if I were not a doctor, no one of them was listening to me and paid no attention. And I thought, well, I just have to get a higher degree. And I started with a master's degree working part-time in the lab and going to school part-time. But the university, first of all, the doctors still didn't listen to me. And secondly, the university would not allow me to go for a doctorate part-time. I had to quit work and go back to school. So that's what I did. And I got a bit waylaid by academics and sort of forgot about the clinical laboratory aspect of it, which is what I loved, until a microbiologist named Dr. Ray Kaplan, who is still practicing. He's a clinical microbiologist directing one of the really large reference laboratories in the United States. He came and gave an academic seminar at the University of Wisconsin talking about his work with Campylobacter in monkeys, which I believe at the time was called called some other organism. I can't even, maybe Helicobacter. And he reminded me about how much I loved clinical microbiology and the bench aspects of it. And he also told me about the American Society for Microbiology postdoctoral educational programs in clinical diagnostic micro, which I had no idea existed. And so I credit Ray with getting me back excited about clinical microbiology and wanting to do a clinical micro postdoc as opposed to a strictly academic postdoc.
Oh, so after your PhD, you went on to do a clinical micro postdoc. Where did you do that?
I did that at the Wadsworth VA and UCLA in Los Angeles, California.
And I'm familiar with traditional, research postdocs. What do you do in a clinical micro postdoc?
Well, I did something slightly different from what normally you do. So a normal clinical postdoc for a PhD fresh out of academics would be to spend 2 years in the laboratory, primarily learning the bench microbiology and virology laboratory techniques that one uses and how to interact with physicians, but not as much physician time spent as laboratory communication with physicians, etc. But because I had come into the field after having been a laboratory microbiologist for 12 years, I didn't need to spend all that time on the bench. So I was free to do infectious disease rounds with the doctors. And so for 2 years, in addition to helping in the laboratory and learning new things, and of course, learning how to implement tests and all the activities a lab director needs to know about, such as protecting the safety of workers and regulations, and nowadays bioterrorism, organism identification and management and budgeting. I also learned a lot of infectious diseases working with some incredible physicians at the UCLA system, doing patient rounds for 2 years.
So is that something that a clinical microbiologist does, is to go on rounds with physicians?
I think they still do it a bit, but I don't think they do it at all to the extent that I did it, because I didn't really need that much laboratory time. I was doing okay in the lab.
I see from your CV over at Stanford that you were director of the clinical lab at Stanford for 10 years.
About 15 years.
15 years. But that wasn't right after your postdoc, was it?
Oh, no. No, right after my postdoc, my first job was in New York. So I had culture shock moving I grew up in South Dakota. So from South Dakota to California, not too strange, and Wisconsin, of course, was more Midwestern. But then moving to New York, that was a shock. And I was the micro lab director at North Shore University Hospital on Long Island for 5 years. So that was my very first job. And that was a very exciting time in clinical microbiology because HIV/AIDS was just being characterized. We started it at UCLA, and some of the very first ever recognitions, some famous HIV doctors like Michael Gottlieb were there at the time. And then I moved to New York, and I worked with Dr. Mark Kaplan, and he had been hand carrying tissues from what we were then calling gay-related immunodeficiency patients, GRID, hand carrying those tissues down to Bob Gallo's lab on the airplane because Bob needed a source of tissues to try to isolate what he was certain was a virus.
So it's not something you'd do today, right?
Not exactly. No.
So you were you were director at the North Shore, is that correct?
Of the micro lab, yeah.
So what is it in a micro lab? Let's get an idea of how a clinical micro lab functions. There's a director, and then what are the others?
Not all laboratories have directors. I would say in the United States, there are about 2,000 laboratories that are in hospitals, and there are tens of thousands that are not in hospitals, that are in doctors' offices. We call them, for example, physician office labs, POLs. But there are about 2,000 hospital and large reference laboratories. Many of them have doctoral level directors. I would say about 300 or 400, but the rest of them don't. They have pathologists who are the directors of the laboratory and have some oversight for the microbiology area. But I think those laboratories that do have a microbiology trained doctoral level director, whether they're a PhD or an MD, have shown in a paper that I wrote actually a number of years ago, higher satisfaction ratings by the infectious disease doctors for the performance of laboratory diagnosis.
There are also commercial laboratories, right, that do this as well. How many roughly of those? Do we have any idea?
Oh, there is at least one or two in every state.
Yeah.
But many of them are owned by two or three big companies.
Right.
Quest and LabCorp and
right.
Yeah.
And so they would have directors, I guess, as well, right?
And I would guess they have doctoral level directors in several sections of the lab. Many labs are broken up into virology and microbiology, or traditional bacteriology, mycology, parasitology.
Right.
And some labs, Mayo Clinic, for example, has doctoral level directors in many different sections.
So what are the duties of the director of these labs?
The duties are to, first of all, create a menu of tests that are appropriate for the patient care needs of those patients that utilize that laboratory service, and to make sure that the tests are done with high quality and reliability, to make sure that the people who are doing the testing in the laboratory are properly licensed if necessary, or accredited and competent. So they do annual competency assessments of the people doing the testing. Directors usually have total oversight for proficiency testing, laboratory safety, making sure that the people who work in the lab are doing things in a safe way so that they don't harm themselves or each other, and then that the patient results are delivered in a timely way and make a difference in the patient's care.
So the director does not do any testing him or herself, right?
Oh, that depends.
Really?
Little labs, a director could actually sit down and do some tests. I certainly did tests as a lab director in some of the labs I worked in, sometimes a gram stain would come in that would be a stat, and everybody else is pretty much busy with culture results and working on things, and you as the director could just easily sit right down and perform that gram stain.
Right.
Sure.
Cool.
Directors often sit at the bench and do work.
It adds to morale too to see the director come out of their office and actually do things. I know our lab director here at the Medical University of South Carolina is Dr. Lisa Steed, and she does bench level rounds with her technicians almost every day. And, she gets in there and rolls up her sleeves, and it's amazing to watch how much hands-on experience she actually takes in training pathology residents, medical students, and really brings to the table a whole new level of richness that you just don't get from attending lecture. One of the things, Vincent, you may or may not know is that most medical schools have eliminated the laboratory component of microbiology.
Sure.
I mean, it's a rare event that medical students today learn how to gram stain.
Yeah.
It's just been phased out. And so what our med students get to experience is they rotate through Lisa's lab in small groups, and she actually gets to show them how a clinical lab operates. It's just not an order sheet that you say, I want this test, and she really instills to them the importance of specimens, and where you take the sample is important, and how you take the sample, and transport medium, and so it's really a great service that many of these laboratory directors do for the education of future healthcare professionals.
Yeah.
Well, thank you, Michael. That was something that is not a general thing, but at a university or an academic medical center lab, probably the largest component of the lab director's job is the teaching component. So you teach medical students, pathology residents, infectious disease fellows, and a lot of other people. And laboratory in-lab rounds, I think, are absolutely critical. And at Stanford, we're going backwards with our medical school training program, and we're revamping it. I'm hoping it will be a standard for the whole U.S., and we're actually going to bring the laboratory back into the basic medical school training curriculum. And not only their own lab, which we do have still a microbiology lab that's hands-on for students in the med school, but we're going to actually, if I have my way, make medical students be responsible for a lab test in each of 3 or 4 different lab divisions, such as chemistry, hematology, blood bank, and micro, and that they will be at the patient side, either collecting the sample themselves or seeing it collected, hand carrying it to the laboratory along with whoever normally would do that, and then being in the laboratory with the technologist side by side as that result gets created and then delivered back to the patient, where then they can implement the patient results. And I'm hoping to bring that into our training program starting next year, because you're absolutely right. Physicians today see the laboratory as a giant black box with something magic happens inside, and the next day they get the result, and they have no clue about how important the sample is, the collection of it, free from contamination, etc. So I think teaching is really important.
Well, one of the great things about TWiM is I'm going to actually use that recorded announcement to take to our curriculum committee, because I've been one of the strong advocates of really arguing for the importance of understanding how the sausage is made, if you will.
Yeah.
And one of the emerging things that we see in our discipline today is the emergence and importance of the microbiome, where sampling is going to become much more commonplace in the family physician's office, because the microbiome seems to be responsible for, you open the morning newspaper and you see yet again a microbe is in charge of doing something else. And so So I think what we're going to see is as we begin to train the next generation of clinicians, we really need to instill in them the importance of proper sampling, how the tests are done, how the question is phrased. And unless you're trained at an early age to appreciate the wonder of the microbes, you're just going to think it's a send-out event.
In many hospitals, it's becoming a send-out event because they're combining laboratories and for efficiencies. At Stanford, our micro lab is actually 4 miles away from the hospital. And so when I was working there for the first 12 years, we had rounds every day in the laboratory, which were tremendously valuable to the physicians. We discussed individual patient cases, and I'm sure important clinical decisions were made on a daily basis, such as you described that Lisa's working in still. But when the lab moved off-site, those daily rounds disappeared. And now the laboratory director, Dr. Benai, and Dr. Pinsky, the virology lab director, collect samples all week long. And once a week, they drive over to Stanford with a box full of stuff and hand it around and have a 1 to 2 hour didactic session. But I don't think that takes the place of daily interaction with real patient samples in a real-time fashion.
No, and oftentimes we see our attending physicians will go down to the laboratory and discuss a complex case with Dr. Steed, and it really adds a lot to that very complicated case that is just not something simple like a garden variety urinary tract infection that is seemingly trivial if it's only E. coli.
Exactly.
So Ellen Jo, aside from the PhD director, the other members of a clinical lab, would they mostly be bachelor's and master's level, or are there some PhDs in there as well?
It depends on the size of the laboratory, but if there are other PhDs or MD pathologists that are clinical laboratory trained, nowadays we have a lot of MDs who go through their pathology training called CP only, clinical pathology only, and they have a career path that is research and laboratory oriented as opposed to tissue pathology oriented. And so they'll come into the laboratory and be directors. So the director of our virology lab at Stanford is an MD PhD. And so he's an MD pathologist with a PhD as well. So we see MDs coming into as lab directors more and more these days, as opposed to just PhDs that we used to have about 10 years ago. Mm-hmm. But most of the workers are bachelor's degrees with medical technology licenses or specialists in microbiology. But that specialist in microbiology, those numbers are not that great. They're mainly bachelor's degree med techs or clinical laboratory scientists, which is the current term, CLS.
So give us a sense for the size of a clinical lab. I know there's a big range, but big ones versus small ones, how many people are working in them?
In a whole clinical laboratory or just the clinical microbiology?
Let's just say the microbiology part.
Okay, so I would guess Stanford is one of the larger, and we have almost 60 people working in the two areas. I might add that we have non-bachelor's degree people. There are medical laboratory technicians now, which could be a 3-year degree or a 2-year degree plus apprenticeship. Mm-hmm. And you have laboratory assistants who have different training, but not college degrees, who do a lot of tasks in the lab these days on automated instruments and on sample preparation and laboratory accessioning and that sort of thing. So the field is open to all different levels of education, not just PhD or bachelor's degree, but even below that. So we have 60 total. But there are microbiology labs in small hospitals that have 3, with usually a PhD would not be there. But I've seen PhDs directing labs with 10 people.
Yeah.
The other thing to appreciate, Vincent, is many hospitals run 3 shifts. Mm-hmm. They operate 24/7.
I see.
That's for sure. If you've got a blood culture coming positive on a sick patient at 2 a.m. in the morning and it's an organism that was not covered by the empiric therapy, you have to have a trained microbiologist or someone who can read that gram stain from that blood culture in the middle of the night and call a clinician who can react and change the antibiotic. It saves lives to have somebody be there in the middle of the night. It's not universal. Unfortunately, there are laboratories that cannot afford to staff 24/7, but I know those that do, it makes a big difference in their patients.
Wow,
and that's the fundamental problem with moving more and more of the microbiology to send out, because you don't get that instant communication often as much as you would if it was your own laboratory in your own facility.
Is that happening more and more? The send out? Why is that?
Costs.
Yes. It's just budgetary issues. And if you can consolidate services at one site for 3 or 4 different places, it's just an economy of scale.
Mm-hmm. Is the bacteriology and virology typically separated into 2 labs, or they can be the same laboratory?
They can be the same laboratory, but it seems to work better in my experience with 2 different laboratories. It's kind of a different mindset. And nowadays, much of virology is done on molecular instruments. There's very few places that still do traditional cell culture and tissue culture work. And so it's functioning more like a chemistry laboratory these days.
Yeah. When I first came here at Columbia in 1982, of course, at the time they had a clinical virology laboratory in addition to the microbiology lab. And I was told that was unusual to have a separate virology lab. But I guess it's not as unusual any longer.
I really can't tell you. There are a lot of virologists who are working in just the regular micro lab.
Yeah.
And if you go to the ASM now sponsored Clinical Virology Symposium, which just transferred over to ASM hands for the first time this year, there will be hundreds of people there, most of whom are general laboratory directors, part of their function is to run the virology testing in those laboratories. So my guess is that they're combined more often than they're not. But there's still some labs that are totally separate. And ours at Stanford is, Mayo Clinic is, et cetera.
But I guess, Mike, the real question is there always a clinical virology component, or are there some where they can only do bacterial work?
There are many where they're not doing much bacterial work, but there are new instruments available for virology testing, PCR instruments that are moderate complexity that allow anybody in any laboratory, even those that are not licensed, if they're moderate complexity, as long as they have a technical supervisor there, to run those tests. And so I think virology tests traditionally were separate and special because they required such expertise and unusual methods. Now they're coming into the mainstream. And so there are at least 3 or 4 different companies that provide real-time rapid turnaround virology tests, respiratory pathogens, HIV type tests, rapid. They're coming into the forefront, and I think more and more labs will be doing virology every day, whether they used to or not. Enterovirus used to be a very difficult virus to detect in spinal fluid, but there's a real-time genetic instrument that is moderate complexity almost, I mean, except for the pipetting part of it, that can be done very quickly in laboratories.
This is one aspect that you probably have a great sense of, having been in the field so long. So think back when you first went into North Shore. What kind of tests did you do back then? Give us a feel for it, and then tell us how it's changed and what's been added today.
Well, I went to North Shore in 1983, and we were doing a lot of traditional cultures, but we did have a blood culture instrument, but the blood culture instrument was using a radio active indicator of growth. So we still have blood culture instruments, and it was quite manual. You had to load bottles into things and carry them, and you had to carry the bottles to the incubator and then take them back to the machine to detect the growth on them. So it was a very early kind of blood culture instrument, but at least it was not manual. When I first started training, we didn't even have instruments for blood cultures, but we had that. But nowadays, you put your blood culture bottles into an instrument. It has automatic shaking in there, and then it has a detector that detects signs of growth every 10 minutes. And so you can detect positives as soon as they occur, as opposed to only doing it once a day when you have the time to run your giant rack of bottles over to the machine.
And think about it, Vincent. The way those old blood culture machines worked is they were looking for evolution of radioactive C14 CO2. And if you haven't seen one of those machines up close, they're really a marvel, especially when you consider that the way the new machines work is you have aerobic blood cultures and you have anaerobic blood cultures. And the machine tells you which one is which.
But when you were using the radioactive growth, did that tell you what was growing?
No, and neither do the ones today tell you what is growing. Although today, if you have a positive blood culture, you can do a molecular test on the broth of that blood culture, and within an hour, you can know what it is. Whereas in those days, you would do a gram stain, and then you would make a subculture, and you would wait the next day till it had a colony, and then you would wait another day to do susceptibility tests. Nowadays, you could do a detection of a Staph aureus and methicillin resistance all within an hour of the machine signaling you that it's positive.
Is this based on PCR largely?
Yeah, based on PCR, or nowadays there are laboratories doing MALDI-TOF, matrix-assisted laser desorption ionization time of flight, MALDI-TOF detection of and identification of organisms from blood culture broths in about 15 minutes of preparation, and then you toss it on the machine, and within a half hour you know the genus and species, sometimes even the strain of organism that you had growing in there.
Wow.
And that test is, if you factor out the cost of the instrument, that test is principally a dime. It literally can be done for 10 cents if you take out the cost of the machine. And when I was in graduate school in the early 80s, those mass spectrometers were, the size of your modern research laboratory. They occupied the whole space. And now the MALDI-TOF machines, which is even more sophisticated than the mass spec machines than we were tinkering with as lowly graduate students, are now more sophisticated, and they're much, much smaller. And it really shows how the clinical microbiologists have really taken what we were doing at the bench and really have transmitted it to a bedside application where you can get real data that before would take 3-4 days. And now, if it's done in concert with the blood culture machine, you can have data pretty quickly. And they're getting more and more sophisticated. Right now, the current generation, and Ellen Joe, correct me if I'm wrong, is relying on a single colony, but more and more they're dealing with samples that are of greater complexity.
Well, I think the electron spray ionization modification of mass spec is the one that you're referring to, that can deal with mixed cultures or initial samples and identify. That one is a little bit more, it's not a dime, and it's about a 6-hour, 8-hour process using a $700,000 machine. So, but MALDI-TOF can go direct from a blood culture broth with a little bit of sample prep to an identification, at least, of a single organism and off a colony, of course. But MALDI-TOF as itself does require an isolated colony. But that, I believe, is going to be the way of the future for many laboratories. Nowadays in the US, we see probably 10 of them around, maybe 20. Europe, they're almost universally used for identification of organisms. So as the licensed and experienced medical technologists, microbiologists retire, the average age in labs in the United States is in the 50s. So they're all going to retire sooner or later. We don't see quite so many people moving into that pipeline. I think MALDI is going to be one way that labs leverage the people that they have, so that instead of doing biochemicals or putting instruments onto biochemical microtiter plates and waiting 6-8 hours for the organisms to grow and metabolize and give us the genus and species, We're going to be doing MALDI to identify them, so the answers will be available in minutes. And then I think there will be rapid methods for detection of key resistance factors, such as the carbopenomases that you were talking about earlier, Michael, or MECA, the methicillin resistance factor, or vancomycin resistance in Anaerococcus, etc. So I think those are the changes that have made and will be making the biggest difference in the practice of microbiology from when I first started. And we identified everything biochemically, taking days, sometimes longer. Anaerobes, my God.
Well, that's what I was just going to ask you about. When once upon a time, when you started out, every microbiology lab had an anaerobic chamber. But
they still have chambers. You got to get those things to grow. But identifying them you can use jars. That's right. We didn't have jars in the early days that worked so well. Now we have machines that will bring down the atmosphere in the jar, so you no longer have to add water to some kind of a catalyst packet and stick it in there and wait 2 hours for the oxygen to be destroyed. But you can bring it right down to vacuum and put in your other gas very easily with the machine. But the identification of the anaerobes has changed a lot. We did a lot of biochemicals and pre-reduced anaerobically sterilized biochemicals and gas liquid chromatography patterns to identify the anaerobes. And in my lab at Stanford now, if you got an anaerobe that you didn't identify immediately from some spot tests and just colony recognition, we would sequence it. So we'll just roll it into a sequence program. And we've got molecular technologists who rotate through molecular in the microlab 2 or 3 every day. And they're sequencing bugs that we don't know what they are. If we had MALDI-TOF, we'd throw it on a MALDI, and they're just starting to look at that there.
Wow. So in addition to blood samples, which I guess is a very common thing to get in a clinical microlab, what other kinds of specimens would you look at?
Well, you get tissue samples from surgery, and you get aspirates from abscesses and subcutaneous infectious areas. You get bone biopsies from the orthopedic physicians who are thinking about osteomyelitis. You get spinal fluids when patients are suspected of meningitis or encephalitis. You get sputum from patients where they're concerned about pneumonia. You get bronchoalveolar lavages or other more invasive respiratory samples if the patient can't spontaneously expectorate. You get stool for parasites and stool for pathogens of all sorts, viruses and bacteria. You get throat swabs and swabs from wounds, and urine for urinary tract infections, and just about anything on a person can become infected. You get hair even.
I was just going to say, how about
fungi.
hair?
Absolutely. They're, the agents of fungal diseases, ringworm and all that, skin scrapings, you name it.
All these specimens, can they be analyzed by MALDI-TOF, or do you have to do other things?
Well, MALDI-TOF at this moment requires about 10 to the 7th organisms per milliliter to identify it. And so right now, most people are not using it for direct samples. But my guess is that people are working on trying to make it work better. But PCR is being used for direct samples, and that's working extremely well. So there's PCR tests for spinal fluid, for enteroviruses, and also for agents of meningitis. There's just been FDA cleared a PCR for stool, which detects something like 20 different agents of gastroenteritis, including viruses and parasites and bacteria, E. coli, Shigella-like toxin, Shiga toxin producing E. coli's and Shiga and Salmonella and Vibrio, cholera and norovirus and you name it.
Wow. So in addition to, so we have PCR and MALDI-TOF, those sound like pretty modern developments. Yeah. Are there any others that we're missing?
Well, there's a PCR-like technology called LAMP, loop mediated amplification, which it amplifies DNA, but it happens at only one temperature. You don't need to do heating and cooling like you do for PCR. It happens at only one, like around 60 degrees. And there are some tests out there, commercial products, FDA cleared, using that technology for detecting, for example, Clostridium difficile in stool. I mean, it depends on the technology and what the manufacturer determines would be a good market for them to use.
Mm-hmm.
There's a Gene Expert platform from Cepheid that has a cartridge format for PCR that will take direct samples of many sorts. It will take swabs for group B strep and stool swabs for C. difficile and swabs from wounds for Staph aureus and nasal screening for MRSA, and all kinds of different sample types can be used in that cartridge.
Mm-hmm. And don't forget the one for TB, which is really pretty terrific because nobody has figured out how to make TB grow any faster. And
exactly. So that TB test is transforming the world. It's over 1 million tests have now been done. And in South Africa, which has deployed it widely, they have found twice as many patients during their initial visits positive for TB as using their previous technology. That technology, which is the acid-fast smear, was 100 years old and hadn't changed in 100 years before the introduction of the Gene Expert multiplex. It's kind of a nested PCR in a cartridge, direct from sputum TB test. And that one is very exciting because of the implications with low resource countries suddenly being able to do the most advanced TB test in the world. As a matter of fact, the only country in the world where the test is not able to be performed is the U.S. because it has not gone through the FDA process yet.
Ellen Jo, when I was trying to schedule you for TWiM, I learned that you travel all the time. Do you do that to teach people clinical microbiology?
I have 3 jobs. So Stanford is one, and I do still teach at Stanford and still consult to the clinical labs, but I'm no longer directly responsible for them. I'm the director of medical affairs at Cepheid, which is the company that makes that TB test that we were just talking about. And so for Cepheid, with all of our 14 different tests that we have, I do a lot of travel around the world, going to meetings, TB meetings, sexually transmitted infection meetings, because we have a GC chlamydia test, and many places I train them how to use it, but I explain to them about the literature and what's been published on it and how the system works, and help people to understand it. And then I have an NGO called Diagnostic Microbiology Development Program, DMDP, which does laboratory capacity development in developing world. And so when I go to give my basic micro workshops, so it's sort of a funny dichotomy. For Cepheid, I'm talking about the most advanced kind of molecular microbiology there is. And for my NGO, I'm talking about the most basic kind of microbiology there is. We're back to teaching people how to do a urine culture and how to read a gram stain. And so for that NGO, I do training in Africa, Southeast Asia, Central South America, etc.
I can imagine it must be very difficult in many of those places to do these simple tests. Do you think you can teach them and they can do it competently?
What we have found, first of all, is that their training is nowhere near as good or as long as what we're comfortable with here in the U.S. So they come to the training that I present with a much less experience and less knowledge background. So that makes it difficult. So what I found is that after an initial week of basic training and implementation of the flowchart program that we use, I'd like to see a volunteer from the U.S. or somewhere who's an experienced microbiology work with the laboratory group for 2 years, hand in hand, day in, day out, until they feel comfortable to be able to continue on their own. And we've found for developing world that it takes 2 years of constant supervision, encouragement, etc. Part of the problem is that the culture of patient care, the culture of quality is just not there in developing world. And we have to mentor them and demonstrate that and try to change the attitude. And the second problem, I believe, is that the governments, we only work in government labs. We don't work in sponsored labs. And the governments never pay the workers enough money. So everywhere they have to have a second job or a third job that does pay enough for them to live. So their first job where we're trying to train them is not their first priority. And getting them to come in on a weekend to read an important culture or staying late, they've got their other job to go to. And if they don't go there, they don't eat. And so it's a very different way of looking at training.
So earlier in our conversation, you mentioned that clinical microbiology was a good profession. There's job security. So maybe we can give some advice to people in their earliest parts of their careers who are thinking about going into the field. What should they do?
Well, I think the first thing they should do is go visit a local clinical microbiology laboratory, talk to the director or the supervisor there, see if they could come in and do some volunteer work, maybe on a weekend, help with sample processing or even just filing reports, something they could do so they could be in the environment and see how they feel. Many people are squeamish about looking at sputum or blood or feces, and if that's their problem, this is not their career.
Plus the smell. I've met many fine clinical microbiologists who, when they go to pick a colony off of a plate, can almost identify to the species what's coming off just based on smell.
Yes, a good sense of smell is critical, but we do have rules now. I've written some papers on this, not to open up certain plates, and because you can acquire, although I don't believe too much from jumping the organism off the plate into your nose, but it is a risk, and so you tend to want to smell carefully, and only once you've ruled out certain bioterrorist organisms and
not even bioterrorism, and just consider the tragic case of Neisseria meningitidis that you saw out in your area with the laboratorian who is working with the Neisseria and unfortunately caught the microbe. And
how did he do that, Michael? What happened?
This was a technician in a research lab out in San Francisco at the San Francisco VA, and they were trying, if I'm remembering correctly, trying to develop a vaccine candidate for Neisseria meningitidis, and this individual was working with it on the bench, and unfortunately, Neisseria meningitidis, the hallmark is that this is a lightning-like infection. It literally strikes you like lightning, and this individual went to bed feeling unwell, and he was literally dead, unfortunately, the next morning. And it was a laboratory accident when he should have been working with the agent in a biosafety cabinet, and he was working with it on a bench. And I remember discussing it with Dr. Steed at the time that it first hit the press, and Lisa said the great thing about the internet is that all the clinical laboratorians talk amongst themselves, and what they've appreciated over the years with isolating Neisseria from a patient's sample, sometimes the patient survives, sometimes the patient doesn't, and when the laboratorian now routinely sees gram-negative diplococci that come off of that initial gram stain, they immediately tag the plate, they seal it with parafilm, alerting the next laboratorian that has to do anything with that particular culture of the potential risk associated with that plate, because just picking and streaking, you can generate an aerosol, and the organism, you don't need very many, it appears, in order to become ill. So they've instituted a practice where all work with gram-negative diplococci immediately go to the biosafety cabinet, where that risk will be minimized. It's never completely eliminated.
So, Ellen, Joe, back to the lab. So if someone visits and they're comfortable with the clinical smells and all of that, what do they do next?
Well, then they would start taking courses in college that would allow them to become a laboratory person. It depends. If they wanted to be a medical technologist, most universities have a medical technology school where the appropriate courses are there. If they want to be a PhD microbiologist, they wouldn't need to be taking the same kinds of classes, because medical technologists are generalists in the laboratory. They have to have as much information and knowledge of chemistry and hematology as they do microbiology. But if someone were really microbiology focused, they could take a lot of science, physics, math, microbiology, and then go on to grad school in any area of microbiology, and then later do a clinical microbiology postdoctoral fellowship,
Right.
such as the CPAP programs that ASM sponsors. And then they would be qualified to at least start out as an assistant director or an associate director in a lab. In a few more years, they would have enough experience to take the American Board of Medical Microbiology diplomat exam, which is something that we advocate from ASM as a sort of a certificate that shows that you have mastered all those areas of competency that it takes to be a lab director. Public health knowledge, such as what we've just heard about with the Neisseria meningitis issue and all those sorts of things, lab safety. Mm-hmm. And then you would become a diplomat certified person and you would continue to direct a laboratory, public health, or reference laboratory, or hospital clinical laboratory.
Are there PhD programs in clinical microbiology per se?
I don't think so. Yeah. If there are very few. Maybe University of Minnesota may have one, but I think most PhD microbiologists do a sort of an academic program. Yeah.
Right. Our colleague Elio, who couldn't be here today, I don't know if you saw a few weeks ago on his blog, Small Things Considered, he wrote a post called The Excitement of Clinical Microbiology. He went to visit the local lab near him, and he wrote at the end something I wanted to ask you about. He said, even in a high-tech world, human skills will still be required.
Yes, I read his blog, and I thought that was great that he appreciated that, because we're still growing the organisms on plates. We're still doing a gram stain. Despite the fact that we have these incredible molecular methods, the cheapest, fastest, easiest way to determine, number one, if there's an infectious process going on at all in the sample you receive in the lab, and number two, an early hint as to what the etiological agent might be, is to stain it with gram stain reagents and look at it under the microscope and say, hey, what is this? And you can call the physician in 20, 30 minutes and diagnose all kinds of diseases immediately. And everything else is just confirmatory after that. And that requires a lot of time and knowledge and expertise to be able to accurately interpret gram stains. And then once you grow those organisms on the Petri dishes, we use many different kinds of Petri dishes with different nutrient supplements and different colored augers and dyes and toxins and antibiotics. And just to recognize, as we were talking about earlier, being able to smell or pick the colonies from the way they look and how they hemolyze the blood cells in the blood agar plate, all those things take a long time to learn and master and feel comfortable with. The hardest part that I find training the microbiologists in developing world is on the Petri dish, when the colonies are all there, teaching them to differentiate between the ones that are just normal colonizing organisms on mucous membranes that are not pathogens and the ones that are likely to be pathogenic in that particular site. That's something that takes a long time to figure out as a bench level microbiologist. And that's totally different from virology, which, as I said to you, was not so visual anymore, except for direct fluorescent antibody reading. And that's another thing that takes years of skill and training. At Stanford, we have 2 reads for every DFA direct fluorescent antibody slide that we do. One person reads it and a second person checks to make sure that the first person's interpretation was what they would agree with.
Must drive you crazy when you spend years training people and then they go somewhere else, right?
Yeah, that's what happens. So it certainly happens in the developing world. All of our good people get snapped up by somebody who pays them better.
Sure. Sounds like a great place to work.
Yes, I think microbiology and infectious diseases are just as exciting as anything one could think of, and they're visually beautiful. I am an aesthetic person myself, and so the colors and the shapes are very lovely to me. It's one of the reasons I gravitated toward this field, is because it's visually interesting and exciting. And it takes a lot of decision-making skills. It's not just putting a sample into an instrument and getting a number out and saying, okay, it's 3 or it's 10. There are nuances and interpretations that require your brain. So I think it's a great field for someone who wants to think and make up their own mind and contribute to the world.
It's just unfortunate, Vincent, that we don't have a CSI-like show to highlight the wonder that is clinical microbiology, because it's so much cooler than looking at a gel that's giving you a particular ladder pattern. Because if you just look at Elio's post on the clinical microbiology, he's got these beautiful pictures of strep pneumo and strep pyogens growing. He's got EM shots. He's got colorized shots. And then he's got, a simple blood agar plate with hemolysis from a strep throat that, each and every one of us has at least been worried about having at least once in our lifetime. And we really need a good television show.
Michael, funny you should say that, because when I lived in LA, I wrote a screenplay. I wrote scripts for 5 shows, and I had treatments for 30. Wow. And I shopped it around all of the directors and producers and writers I could find, and there wasn't one of them who thought it would be a good idea. They said, "Ooh, you're talking about feces? You're talking about sputum? No, we don't want to deal with that. We're not interested in that." It was kind of like a mission impossible for microbiologists. I had all the characters fleshed out based on real people that I knew. I thought it would be a fabulous TV show, but nobody would buy it.
Maybe you were before your time. Isn't that what House does now?
Sort of.
Well, House did a lot of microbiology, and House was a very successful television show, albeit he had too many lupus cases.
Yeah, but he had his own dog. He had his residents and interns going down in the lab and doing everything. And of course, they never do that. So It was a little unrealistic.
But I think, it really shows the wonder that is microbiology. And when you talk to the folks in the clinical laboratory, and you quickly appreciate that each day is an adventure for them, because when I talk to Dr. Steed, she always tells me about the coolest thing that she's seen, or she calls me up and says, "You got to come over and see this plate." And so I go over and I look at the plate, and you're just continually amazed at what grows up. And unfortunately, many of these things have come out of patients, and you appreciate the service that they're providing, because the good news is we have medicines that can cure someone literally overnight.
Yes, that's true. One of my first exciting moments was I was looking through a stool, and the proglottids of a tapeworm were in there, and they're actually still moving. And here was this stool sitting in a petri dish inside the hood, and I was sort of straining through other parts of stool because the doctor told me to find the head. And the proglottids that were part of the tail part stood up in the petri dish and started waving. And wow. I had no idea they would do that. I screamed. I was so afraid. It was one of my early formative moments in microbiology.
Wow, that's great. Well, that's a good way to wrap up this episode. Is there anything we missed, Ellen Jo, that we should have talked about?
Boy, I don't think so, but I'm sure Michael has thought of something else.
Anything from you, Michael?
No, I'm good, Vincent.
That was a good one. This episode of TWiM, like all the others, will be found at microbeworld.org/TWiM. And if you like what we do, what you can do to help us is to go on over to iTunes and subscribe, and also leave a rating there. And that helps to keep us visible so more people can find us and hear about this wonderful world of microbiology. If you have any questions or comments, send them to twim@twiv.tv. Ellen Jo Barron is at Stanford University Medical Center. Thanks so much for joining us today. We really appreciate it.
Oh, thank you. It was a great pleasure.
And I hope this gets us going. Certainly has gotten me very interested, and maybe we'll do more clinical microbiology on TWiM in the future.
Sounds like you ought to talk to Lisa.
Yeah, it does sound good right there where you are, Michael.
Oh, yeah. Well, the other person I suggest bringing on for This Week in Parasitology is Lynn Garcia.
Sounds like a good idea. Hook us up.
Great idea.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thank you, Vincent.
Always a pleasure. I'm Vincent Racaniello. You can find me at virology.news. And I want to point everyone to a documentary that we just made together with ASM. It's our tour of the Needle, the high-security biological containment facility in Boston, Massachusetts. You can find that at my blog, virology.ws. Many thanks to ASM for supporting TWiM, in particular Chris Kondayan and Ray Ortega for their technical help. The music you hear at the beginning and end of TWiM is composed and performed by Ronald Jenkins, and you can find his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.